Hi, everyone. It's been a while since we've seen you. Hope you're doing well. As many of you know, we've been collecting blood from our patients with PANS and related illnesses and sharing these precious samples with scientists at Stanford and other universities. I wanted to provide an update so you can see some of the science in action. We couldn't highlight everyone today, but here are just a few of the scientists we are working with. Hope you enjoy these short videos, and we look forward to highlighting more PANS science in the future. Thanks for all your support.
Hello, I'm Betsy Mellons, and I'm a professor of pediatrics at Stanford, and I'm also a board-certified pediatric rheumatologist. But I've spent most of my career here in my laboratory trying to understand what causes inflammatory diseases in children and how to better treat them. I started working on PANS, or my group started working on PANS, about 4 years ago because of Jenny Frankovich's deep conviction that PANS is an inflammatory disease. And so we've been working for the last 4 years to identify signs of inflammation in the blood cells, particularly the immune cells of children with PANS. When we get a blood sample from a child with PANS or a healthy control, we bring it down here to this room that we call the tissue culture room. This room allows us to work with these samples in a sterile environment, particularly using these hoods where bacteria in the air or on people's hands are excluded. On people's hands, they're excluded by the people wearing gloves. If we want to keep the cells alive or even expand them, we move them to here, an incubator, which is set at 37 degrees, which is human body temperature, and that allows the cells to stay happy and healthy. And you can see there are a number of shelves where we keep cells. We've been particularly interested in cells that are driving inflammation and cells that are calming inflammation. And that's because PANS is a remitting and relapsing disease. That is to say, there are flares and then there are improvements. And that led us to hypothesize that there would be cells that drive inflammation as well as cells that try to quiet that inflammation. And we're really excited that we in fact have found both kinds of cells in samples from children with PANS, and this differs from what we see in children who are healthy. Okay. So, in order to really understand what those cells are doing, we need to look at the level of each individual cell and look at what its program is, that is what it is doing. And to do that, we use a special little machine, which I'll show you next. Okay, so this little box, which is, as you can see by the size of this coffee cup, really the size of about a shoebox, is what allows us to learn from each cell what is going on. That is a treasure trove of information at the level of each individual cell. And as I said, we're excited to say that we have found cells in PANS patients that are driving inflammation, cells that we might want to repress with some kind of therapy, but also we found cells that are actually trying to quiet the inflammation. And that is a particularly important discovery because if we learn what the body is trying to do to quiet that inflammation, we can try to replicate that and either make it happen sooner or make it happen at all in children where those cells are not already activated. And so our work to date has, we believe, helped indicate that PANS is an inflammatory disease and that we have now new ideas about how we may be able to quell that inflammation in children who are suffering from PANS. Thanks.
So hello, my name is PJ Utz. I'm a professor of medicine here at Stanford, and I wanted to thank our donors for coming here and virtually, and I'm really excited to show you a little bit about what we're doing. So the backstory for this project is that we've been very interested for decades in studying autoantibodies. These are molecules that are part of autoimmune diseases. Many of them are actually pathogenic. And so we're testing the hypothesis in PANS that some kids, and maybe a large number of these kids, are developing autoantibodies which could actually be causing the disease. So about 3 years ago, right before the pandemic, we were approached by Brian Kabilka's lab. Brian is a famous scientist here. He won a Nobel Prize in 2012. And he had this idea that maybe some of these receptors called G protein coupled receptors might be targets of the immune response in cognitive disorders or psychiatric disorders. So we contacted Jenny, and Dr. Frankovich was fabulous at helping to get us the right samples to be able to study. Brian creates these proteins in his lab in a way that no one else in the entire world can do. So we have access to these molecules that nobody else can test, specifically for neurocognitive and psychiatric disorders. So Jenny provided funding from one of the donors, specifically to hire Seaway. So Seaway is pipetting there in the background. I don't want to interrupt her because she's actually in the middle of an experiment. And the workflow here is that we get the samples from Brian. Seaway and another person I'll tell you about in a minute spend a lot of time working with these proteins, getting them affixed to beads. And then once they're on these beads, we can use them to try to find autoantibodies. Brian has provided 8 or 10 different proteins that we're now working with. Each one of them are super interesting. So what Seaway does is she's taking these proteins and working with them at the bench in these little tiny microtiter plates, transferring just microliter, like way less than a tear amount of fluid. The samples and everything that we study from the PANS patients are actually right behind us. They're stored in these areas here. So there's an enormous liquid nitrogen tank there that houses tens of thousands of samples. You actually have to go up on steps to even get into it. Everything in there is about 150 degrees below zero to keep it intact. Once Seaway then makes the arrays, she probes them using the samples from the PANS patients as well as carefully collected healthy controls, and then they go over in this direction. So let me now show you 2 of the instruments that we use specifically for this purpose. The first one is this washing dispenser. We got a large grant from the National Institutes of Health to be able to develop this, so we were able to leverage the philanthropy that you're providing. This allows us to make custom arrays so that each of these little tiny beads has a different protein on it. So Seaway makes the arrays here, goes back to the other room where I was just showing you, and then she will add the patient samples, do all the wash steps back here. And from there, they go into this instrument, which is called a FlexMap instrument, also provided by the National Institutes of Health. And so the samples go into, this actually pops out, the samples go in there in these microtiter plates. Like this one, we can do 384 samples at a time. And Seaway has already run about 80 or 90 PANS samples provided by Jenny's group. And then they go through this device. It measures how bright the signal is for each one of the beads, and that tells us what the protein targets, potential protein targets are in these kids. And all that is then stored on a computer. So just to summarize, the goal of the studies here is for us to try to profile autoantibodies in the PANS patients. We're looking at literally hundreds of different proteins at a time. And then that's using traditional arrays that my lab has developed over the last 20 years. But we now have these super interesting arrays that we've developed with Brian's lab that nobody else in the world possibly could ever make because the proteins are so difficult to work with. The last thing I'll say is that we're really fortunate as well that Stanford is a really great training environment for physician scientists. And so we have a psychiatry resident named Tyler Prestwood, who unfortunately had to just leave to go to clinic. Tyler has an MD and a PhD from Stanford. He was in Ed Engelman's lab for his PhD studies. And he has been working in the lab now for about the last year and a half, part-time when he's not seeing patients. And the residency program pays for him. Starting July 1st, he will be full-time in the lab, fully funded by the residency program. So he's been teaching and training Siwei, who is fresh out of college. She's very good, but he's teaching her how to be a good technician. And in terms of philanthropy, it doesn't get better because you're getting an MD, PhD trainee completely free to be able to work on this. And his goal is to build his entire career studying the interface between the immune system and neuropsychiatric disorders, and asking questions like what's wrong with the immune system, the kids with PANS, why they're having these experiencing these terrible symptoms. So thanks again for your philanthropy. We really appreciate it, and I'm glad to open my lab up to these sorts of studies that are just super exciting. And while related to what my lab is doing, they enrich what we're doing by bringing in entirely new questions and ideas. Thanks again.
Well, welcome to our lab space. I'm Dave Lewis, and this is Viswanath Gandhi, and we're very excited about doing research onto the mechanisms of disease in PANS. And we think that it's like a lot of detective work involving the immune system. There are many potential players and ways that things can go wrong so that the body's immune system actually ends up causing disease. And we're very interested in studying the blood of PANS patients because they contain a good number of the white blood cells that potentially can cause disease. And we have a new technology that we would like to bring on the problem that's called cytometry by time of flight, or CyTOF for short, so it's kind of a mouthful. But this is a special instrument that allows us to look at the behavior of literally tens of thousands of different types of white blood cells in the that are in the bloodstream, and to see if we can get patterns that are distinct for patients that have PANS. And this technology is really only about 10 years old and was really developed at Stanford by Dr. Gary Nolan, who's just down the hall, and I'm happy to show you an instrument. But the basic idea is we take blood from patients that we've stored in liquid nitrogen so that it's ready for us to use to do the analysis. We then take out the cells, warm them up to room temperature so that they're nice and functional, and we then stain them with what are called monoclonal antibodies to look at the function of the cells, what type of cells they are, and then run them on this special instrument. And the instrument allows us to look at 45 different properties of each cell as it goes through the instrument, which is the highest resolution that we currently have to do this kind of a study. And we're very confident that this is going to give us new clues as to both the origin of the disease and probably the treatment as well. We're very focused on the function of these cells in patients that have PANS versus some of their siblings and other children their age that don't have the disease. So that's really the long and short of what we're excited about. So I'm not doing this alone. I'm involving Dr. Brice Godelier, who trained in the Nolan lab and now has his own lab, and we've collaborated on all of our studies using the CyTOF instrument. And so we've published a number of papers together, and I look very much forward to extending this to the patients with PANS. All right, we're back. This is a CyTOF 2 instrument, and has a way for the cells that have been stained for the 45 different parameters are taken up, and then it's analyzed by something called a mass spec that allows us to look at, for all of those 45 different things, how much is on each cell. at the same time. And it's really a fairly small instrument. The original versions were much, much larger, but it's now become something that is fairly routine for analysis. And this instrument over here is a MIBI, which is a similar kind of instrument as far as the kinds of things we can look at, but it's actually meant for tissue, not for cells, but actual tissue state. So that's something that could be done later depending on the results we get from CyTOF. But our main focus is going to be on blood cells using this instrument.